restart. Yeah, just briefly. Yeah, sorry about that.
No problem. Okay, so I will. Okay, so the history, the history in the Soviet Union. So I think that the history of infra-low frequencies started in the Soviet Union, and in the middle of the 20th century. And this was time when appropriate devices such as chopper amplifiers for recording infra-slow potentials were designed. And I think that the pioneers, at least one of the pioneers in this field was Nina Alajalova from the Institute of Psychology in Moscow. And I tried to find out the photograph of this woman, and I was not able to find it. Alajalova worked at the Institute of Psychology in Moscow. But there was also another woman who worked in Leningrad. In those days, St. Petersburg was called Leningrad after Lenin. And this woman was Valentina Ilyukhina, and she worked at the Institute of Experimental Medicine in Leningrad. And actually, this was the laboratory where I worked in 70s. So in 70s, I was a postgraduate student of St. Petersburg, of Leningrad State University, with a major in quantum physics. So I was a theoretical physicist, and I was invited by Professor Bektereva, who was the director of the Institute of Experimental Medicine in those days. So I was invited by her to join her team to study the human brain. And they had a new method of implanting electrodes to the patient's brain. And they actually dealt with the different types of diseases like Parkinsonian disease, like obsessive compulsive disorder, like central pain, etc., etc. So they implanted electrodes for diagnosis and therapy. And we got a unique opportunity of recording different physiological parameters from these electrodes. And one of these parameters was, of course, electrical potentials. Although I have to notice that the electrodes were gold, so they were polarizable electrodes. And basically, when you record the potentials from these polarizable electrodes, you record so-called redox potentials. And the redox potentials are actually defined by the next equation. Well, I'm not going into details, but this on the left, you can see the photograph of this woman. She is a professor, and she's still alive. Alajalova died in 1991, and so these two women, they found two types of infra-slow oscillation in the human brain, and with periods of 10 and 30, 90 seconds respectively. So they were talking about periods, about infra-slow frequency, and they were talking about so-called infra-low control system of the brain. Okay, and they published quite a lot of papers. In the same time, I was a newcomer, a kind of PhD student, and I worked together with Valentin Gretchen, and who actually developed a method of recording infra-slow oscillation in the oxygen. So we used a kind of a polarographic method for recording the oxygen availability in the brain. So basically, we used not potential amplifiers by the current amplifiers, and we applied a voltage minus 0.6 volt to the electrodes, and according to the polarographic method, the current that would flow was proportional to the concentration of oxygen near the electrodes. So basically, we're measuring the oxygen availability in the brain, in different parts of the brain. And what we found is that the level of oxygen is not stable in the brain, but fluctuates with the frequency in this low frequency band. So the period of this oscillation was about 10, maybe 30 seconds. So the frequency is about 0.1 hertz and below. And so this was happening in the Soviet Union. And my first book that I published with Valentin Gretchen, my supervisor in those days, is presenting at the left below, and it's called Slow Non-Electrical Rhythm of the Human Brain. So we were describing different rhythmicities, different type of rhythmicities in oxygen, in the impedance, in the local blood flow that we were able to measure using different types of electrodes from the human brain. And we showed that there is a strong kind of relationship between these oscillations. Okay, so, and one important thing that I discovered was that actually the phases of this oscillation are very important. They have a different functional meaning, like rising phase and falling phase, or increasing, decreasing phases of PO2. They have quite different functional meaning. And I think that the whole my PhD dissertation was about this infra-slow oscillation in the human brain. Okay, then the next, Austria and Germany. So in Austria, research on infra-slow frequencies in the human brain was carried by Herbert Bauer and Giselher Gutmann from Vienna University. And I worked with these guys. I think I've been at the in Vienna University of the 90s and 80s and 90s. Can you imagine? I remember that I went to Vienna, to Austria in 1991, and my country was called the Soviet Union, and the city I was working was called Leningrad. And in 3 months, I went back, and the country was called Russia, and the system was called capitalist system. and the city was called St. Petersburg. Can you imagine? In just a few months, everything changed. Well, anyway, I worked with these guys. With here on the left, at the top, you can see the photograph of Gutmann, and he was the head of laboratory at this university. Liebiggasse 5. And they actually recorded EEG with special amplifiers. So Herbert Bauer was an engineer, and he developed an amplifier with high impedance, high input impedance, and with the corresponding frequency band. And they were recording this infra-slow oscillation in different tasks, and the tasks were mostly cued tasks in which they presenting a warning stimulus and then the trigger stimulus, and they recorded this infra-low EEG before warning and trigger and targets. And of course, they found the slow negative variations, which called CNV wave, contingent negative variation. And they tried to figure out what is the difference between healthy controls and psychiatric patients. And they publish a lot of papers. And simultaneously, there was a group of people working in Germany, and the name was Rockström and Niels Bierbaumer. And here on the left, at the bottom, you can see that there's Brigitte Rockström. And they, I mean that both groups, they develop a kind of the EEG neurofeedback that was based on infra-low frequencies. And basically, they also find out that the waves that are generated by the warning stimulus, the amplitude and the polarity of these waves actually were correlated with the performance. So good performance was connected with the moderate negativities, and the bad performance was correlated with positivities or strong negativities, so a kind of U-shaped correlation. And they try to use this for diagnostic purposes as well as for treatment purposes. And I think that you know that Niels Bierbauer, this group, and Tübingen University developed a method of slow cortical potentials neurofeedback. So they use slow cortical potentials for neurofeedback. And I mentioned the name of this slow potentials, which is called CNV, contingent negative variation. And I have to mention the name of a famous English neurologist, Gray Walter, who actually discovered this waves, and the discovery was made in 1964. And here on the right, I present the figure from this old paper of Gray Walter. I have to tell you that Gray Walter was a great man, and he was a friend of the Soviet Union. He often went to our laboratory, at the Institute of the Experimental Medicine in Leningrad. So I met him personally. He was a nice man. Not only, he was also an engineer and neurologist, and he made a lot of discoveries in this field. And one of his discoveries was, of course, CNV wave. So when you say CNV, contingent negative relation, you also have to think about famous Grey-Walter, who was the first who discovered it. Okay, so the beginning of the 21st century, the interest in infra-slow frequency was renewed by the appearance of commercially available DC-coupled amplifiers. So the amplifiers were on the market, and actually people, they tried to assemble the amplifiers for recording EEG. And that was indeed a breakthrough. As I told you, the Herbert Bauer was an engineer, and he spent a lot of time by developing new types of amplifiers for recording the infra-slow. And now the commercially available amplifiers were on the market. So it was indeed a breakthrough. And the other breakthrough was discovery of infra-slow fluctuation in the bold signal in functional MRI. So they found that the bold signal, which actually a measure of local blood flow, basically, in the brain, so it's not stable and it fluctuates, and the frequency of this fluctuation was quite slow, seconds, decasecond. Okay, and this bold fluctuation correlated within brain regions, and it looked like they reflected specific functional connectivity patterns, and the people started talking about default mode network and several task positive networks, etc., etc. And approximately in this time, the neurofeedback protocols based on parameters of spontaneous EEG, infra-slow frequencies, have been suggested and applied in clinical practice. And I think that I need to mention the name of Othmars, Sue and Siegfried Othmars. And I think also I need to mention the name of Mark Smith. And I have to tell you that several reviews on neuroscience of EEG SF, infra-slow frequency, has been published recently. And me too, I also published a paper on infra-slow frequencies. And one of the authors of the Othmar method is presented in this workshop. His name is Berhard Vandernoot. And he told me that, and I also knew Othmars, Othmars and Siegfried and Sue. And they told me that ELF neurofeedback was discovered accidentally, and it was based on clinical observation. And the current implementation is based on more than 100,000 neurofeedback sessions that were used for optimization of the signal chain. So there is a heavy, solid work behind this method that was discovered by authors. Okay, so here I present a paper that was done by a group of people with Mark Smith and Dirk de Ride. And I think it was the first the first randomized double-blind placebo-controlled study of ESF brain training, and the patient they used, they were food addicted. So, and the idea was that the craving for food was associated with activation of the parietal and the anterior parts of the cingulate cortex, and the idea was to train infra-low frequencies in this part of the cortex, and they used Loretta imaging for computing this kind of virtual EEG, and they were able to demonstrate using this double-blind placebo-controlled study that this neurofeedback works. And here on the right, I present the picture, the figure from this study that demonstrates pre and post treatment parameters, including infra-slow amplitude of the oscillations, and this is a S-Loretta image where they made the localization of the sources of the EEG signals, and on the left, this is in the middle. You see that with the gray hair, with white hair, is Dirk de Ride, and on the left you can see this is Roberta Pasquale Marquis, the developer of Loretta and S-Loretta imaging. Okay, so basically, if we talk about neurofeedback, we have to remember that the neurofeedback, the first neurofeedback was discovered in 60s, and we have to mention the names of Barry Sterman, Joe Camilla, etc., etc. And I think that many of you worked with this conventional neurofeedback. And when we talk about conventional EEG, conventional neurofeedback, we talk about EEG in the frequency band from 0.5 to 30 hertz. And in this band, we're dealing with delta, theta, alpha, beta waves, and you can train these waves separately. You can train ratios of this frequency band coherences, etc., etc. So this is a kind of conventional neurofeedback protocols. And but you can also try to go below 0.1 hertz and train the infra-slow frequencies. And there are two types of neurofeedback: discrete and continuous. The example of discrete neurofeedback is the method that now is used by Niels Bierbaumer and his colleagues in the University of Tübingen, and the continuous neurofeedback that was actually applied by the Osmer's method. Okay, when we work with the conventional neurofeedback, we're using some theory, and the theory tells us that alpha waves associate negatively correlate with cortical metabolism, and beta waves are positively correlate with the cortical metabolism. We have the theory that the performance is associated with the metabolic activity according to the inverted U law. We know that, and using this theoretical background, we can construct a protocol on the basis of EEG, of spontaneous EEG of a subject. We compare the EEG spectrum of the subject with the normative database, and you can find if the subject on the left part of the curve, or if the subject on the right part of the curve, and on the basis of this finding, this comparison with the database, we can suggest a corresponding protocol. So we have a theory, and if you talk about ESF infra-slow frequencies, then basically we don't have a theory, and the rest of my talk will be about how to approach to this theory, how to do it. Okay, so equipment. When we talk about equipment, we talk about amplifiers, DC amplifier. The amplifier should be DC, and I remember I told you that these amplifiers are commercially available. And I personally, although I don't belong to these companies, B-Medics and MISAC, but we work closely with these people. And I personally using the MISAC amplifiers, as well as NeuroAmp amplifiers. NeuroAmp amplifiers produced by B-Medic company, and this MISAC amplifier, they are produced by MISAC company from St. Petersburg. Okay, so this is these are devices that we're using. Okay, so this is as I said, I don't belong to B-Medic, I don't belong to other neurofeedback companies. So here I'm just telling you what I know about these infra-low frequencies neurofeedback from literature. And basically we have two different approaches. One is the ESF method that was developed by Othmers, and behind this method a lot of clinical work. And also we have this ESF Associates company that is headed by Mark Smith. Okay, this is the picture, the slide that was given to me by Berhard Vandernoot, the head of B-Medic group, and it shows you the EEG spectra in the full band EEG spectra in double logarithmic scale. As you can see that this is almost linear function. In double logarithmic scale is almost linear function, and the power spectra density defined as 1 by f, which f frequency in power of A, and this the coefficient is 1.5. So basically, this what we know experimentally from the human brain. Okay. And I think that the method that was developed by Osmer, again, this is a slide that was given to me by Bertrand Bondenot, and the Osmer method that was developed in the amplifiers called SIGNet. And as you can see, it has a position together with the other neurofeedback training, with conventional neurofeedback training, with the slow cortical potential training, and with the connectivity training. So electrodes for ESF EEG recording. First of all, the electrodes must be non-polarizable, okay? Because the polarizable electrode materials such as gold, platinum, tin, steel are coupled in a capacitive manner. Why? Because if you place electrodes into the tissue, including brain tissue, on the skin, the double electrical layer is formed between the metal and electroconductive gel. So, and this double electrical layer, the potential of this double electrical layer is defined by the next equation. And basically what you measure, you are measuring the logarithm of concentration of oxidative and reductive phases of metabolites. So it's biochemistry. And basically, if you want to measure the potentials that are generated on the surface of the brain, then you have to use the non-polarizable electrodes. And one of these electrodes is silver chloride electrodes. And the most suitable electrodes and currently commercially available are reusable sintered contact elements. Okay, and you can buy them, and they are commercially available. And these electrodes are absorbent, and they should be cleaned immediately after use with the distilled water. It's also recommended to soak them in saline solution for up to an hour before use. And you also need to use a special gel for stable recording, and the gel must include chloride as a part. So basically, I think that any gel, including the electric cap gel, gel can be used. You just to add more chloride, you simply can pour some salted water in this gel, and that would be okay. So don't forget about having a chloride as ions in the gel. For example, we are using these caps and this kind of gels that are produced by this company. This is a Russian company which is called MKS MKS company. Okay, and EEGs, ESF are susceptible for contribution for numerous sources. So as we know that the artifact come from eye and head movements, and also there are many artifacts coming from skin, from changes, variation in skin properties. So eye blink and lateral eye movement are not actually big issue anymore, because then we can usually correct this artifact using independent component analysis, ICA, by simply decomposing the EEG into this independent component, finding the corresponding component that are associated with eye blink and lateral eye movements, and simply zeroing these components. So it's a quite easy procedure. But the other types of artifact is not easy to correct. The other thing is the integrity of skin electrodes interface. This contact, skin electrode contact, is crucial for minimizing electrode movements and the galvanic skin's response. Remember the galvanic skin's response artifact? It's also the artifact that are generated by the skin, and they need to be avoided. And the best way to avoid electrode movement artifact is to fix electrodes with collodion. And the best way to avoid this galvanic skin response artifact is to short circuit the skin. Can you imagine? By puncturing the skin under the electrodes. And I remember that when I was working at Vienna University, I remember that when these guys were recording the infra-low frequencies, they usually cut the skin under the electrode. Can you imagine? But they don't use the many electrodes, so it was not a big deal. It just simply cut by a needle or something like that, the skin. So, but when you have 31 or 100 electrodes, that could be a problem. And I don't think that anyone is doing this anymore. So the people, they prefer to deal with the GSR artifact, but not to cut the skin on the electrodes. Okay, so I think that now it's time to show you. I think that it's time to demonstrate you the EEG, what we are talking about, and let me demonstrate. So this is a WinEEG software, and this is software on my computer, and we recently finished a study in which we recorded 31 channel EEG, brown band EEG in healthy controls and in patients. And we were using these amplifiers, DC coupled amplifiers. So I can demonstrate you the infra-low frequency oscillation in healthy and patient's brains. So I open EEG file. So let me see. Let us look at the norms. Okay, let us open this file, for example, this file. So this is, as usual, I select, I think that I need somehow to change this. Okay, so I need to apply the corresponding montage, so 31 channel montage. So this is a montage. This is the conventional EEG. Okay, you can see conventional EEG. And first of all, I need to get rid of this eye blink artifacts. So I select 2 minutes of EEG, then go to analysis. I use this option, artifact correction procedure, and I'm using ICA. So I am decomposing EEG into independent component, and one of these component, like here, corresponds to eye blink. And I think that you can see here, this is eye blink. On the left, this is eye blink artifact in EEG. And the next column is the corrected EEG. And on the right, this is a topography of this of the independent component, and the next column to the left is the tie dynamic of this component. So I select this topography that corresponds to eye blink, and I simply correct eye blink. So you see that now in EEG you don't see any eye blinks. And now I want to look to see what is happening in this infra-low frequency back. You see, in conventional EEG, the frequency is from 0.5 hertz up to 30 hertz. Okay, and I'm using the notch filter of 50 hertz. Okay, and the speed of presenting is 30 millimeters per second. So this is a 1 second interval. Okay, and I want to look what is happening with infra-low frequencies. So I need to change the speed, of course, and this is the speed is about 2 millimeters per second. Okay, then I need to change the lower cut frequency, and with the I'm using the lower cut frequency with the 10-second time constant, and the high cut frequency I'm using 0.5 hertz. Okay, so this is EEG recorded in a healthy subject in infra-low frequency band. Okay, you can see that what this is the signal that you are working with when you are doing this in ESF training. Okay, so basically we can do the same that we are doing in conventional EEG. Remember that in conventional EEG, we are computing EEG spectra, and we can compare the EEG spectra. We can compute the EEG spectra for a lot of healthy subjects. We can compute the grand average spectra, and then using comparison of individual EEG spectra with this normative database, we can tell how much the individual spectra differ from this grand average. Okay, so let us do it. So go to analysis, compute EEG spectra, of course. In neurofeedback, in conventional neurofeedback, in conventional EEG, we are using the method which is called Welch method, that actually simply average compute EEG spectra for some epochs and then average those epochs. But now here we're using this a different method, which is called adaptive multi-taper method by Thompson. I'm not going into detail. Just believe me that this is the method that we need to use for this case. And the extent of 4 seconds, the epoch in conventional EEG, the epoch that we use is 4 seconds. Here I'm using a higher, of course, duration, because we need to look at the lower frequencies. So in this case, I'm using this epoch duration 64 seconds. I choose the fragment, and the subject was doing the continuous performance task. So I click simply OK. Okay, and this is the EEG spectra that I computed for this subject. And this is the EEG spectra for 31 channel of healthy subject, the performing repeated task, continuous performance task. And you can see that there are peaks, and remember the peaks, they correspond to some regularities, oscillations, and in this case, in this particular subject, the oscillation are it's 0.11 hertz. This is the first. So this oscillation mostly concentrated at temporal electrodes, T5, T6 electrodes, and 0.11 hertz frequency. And the other frequency is here, and this is a 0.09 hertz frequency. And this the map of this frequency is quite different from the map of this frequency. So basically, this subject has 2 different frequencies, and with the 2 different frequencies, and with 2 different maps of these frequencies. So basically, you can compute the EEG spectra. We're having here hundreds of healthy subjects. We can easily compute the EEG spectra for each of the subjects. We can average them, and we can compare them with the database. So theoretically. And now let me show you the EEG of a patient. Okay, of a patient. I have here the EEG of patients. Let me see. You see that these are depressed patients, norms. This Parkinson. Yeah. Excuse. Let me show you. To the corresponding montage. This is, I need to correct the eye blinks. I select 2 minutes. Let me see. Not convenient. Okay. So let us correct artifacts. Okay, this we are going to do apply. So I select 2 minutes. I'm applying ICA. This is a raw EEG. This is a corrected EEG. So this is a topography of eye blink. And I click OK. So I corrected eye blinks. And I think that I can and I go to analysis. Oh, I need to change. Of course, I need to change. So I change the speed. I change the low cut frequency. I change the high cut frequency. And this what we get. That what we get in EEG. I think that if you can, I think even by naked eyes, you can see that the pattern of EEG in this particular subject differs from the pattern of EEG of healthy controls. I think that let me show you like this. Okay, look, at the left, this is EEG on patients with schizophrenia. And this is the patient the healthy subject. And I think that you can see the difference. So you see that the amplitude of these oscillations are much higher in the healthy subject, and also there is a kind of dominant frequency, is a 0.1 hertz in the EEG of a healthy subject. And let me compute the EEG spectra for the patients with the schizophrenia. So I go to analysis, EEG spectra, and I'm doing the same. So the epoch length duration is 64 seconds. I'm using this adaptive multi-taper method by Thompson. I click OK. And this is a spectra. And this is a spectrum of a subject. And first of all, you don't see any peaks. There is a kind of, there is no dominant band, dominant oscillation in this case. And this is as you can see, it's quite different from the EEG spectra of the healthy subject. And by the way, we used the Osmer method in our laboratory that was given us by B-Medic company. So it was, you know, as a laboratory in Russia, we have a lot of difficulty with the funding our research, and the B-Medic company actually gave us these amplifiers for free, and that we were able to make a study, quite large study, with patients with depressed moods, with depression, and performing the conventional method that developed by Osbert for this disease. And what we found is that at the beginning, the EEG look almost flat. There is no oscillation. This is a low frequency oscillation in EEG, and the fluctuation is very small in amplitude. But after training, after this neurofeedback training using Osmer method, we saw a statistical increase of the amplitude of this oscillation. Indeed, this method works, and we also compared changes with a kind of placebo control. So we ask subject to perform neurofeedback using heart rate variability. So, and the heart rate variability neurofeedback by feedback did not change the EEG. So the changes in EEG, increasing of amplitude of this oscillation, they were indeed developed by the application of this method, of this specific method. Okay, let's go further on. Okay. So I think that, are you getting the happiness of understanding?
Absolutely.
Yeah. So you see
Yes.
that I showed you, I demonstrate you a signal with which you are working when you are using ESF neurofeedback. That is the signal that you are working, and indeed the signal that somehow depends on the state of the subject. So, and using neurofeedback, you can change the state of the subject. I think that there are now we have quite many kind of research project that clearly demonstrate changes in EEG, in this infra-low frequency EEG after neurofeedback in comparison to placebo effects. Okay, let's go on. Okay, so now let us talk about the infra-slow fluctuation in the brain. Okay, so in the brain, you can measure basically impulse activity of neurons. So you can implant electrodes, micro or macro electrodes, and you can record impulse activity of neurons, and you can record the local field potentials. And here I present what we know about those studies. Of course, these studies are made in animals, in rats, monkeys, And here I demonstrate the areas in the brain in which the recording was made, like in the cortex, in the striatum, in the globus pallidus, in the thalamus, and the brainstem. Okay, and in all these areas of the brain, when you record these either local field potentials or multi-impulse, multi-neuron activity, then you see that there are infra-lower fluctuation everywhere. There are infra-lower fluctuation. And for example, you record EEG in the brainstem, okay, and you compute the power spectrum of a signal unit, and you can see there are quite clear peaks at different frequencies. And also, if you record the thalamic neurons, also you see the same. The infra-low frequencies. So, and think about this, that, for example, brainstem nuclei, like nucleus cerebrus, like Rafe nucleus, they are projected to the cortex, and basically they are in position to generate infra-slow fluctuation on the cortical level. So we're talking about neuromodulatory pathways. And we're talking about noradrenergic, serotonergic, dopaminergic, and cholinergic pathways of the brainstem and the basal forebrain. And they can easily generate this infra-low fluctuation in the cortex. And if we talk about the basal ganglia, which are also projected to the thalamus, and from the thalamus back to the cortex, so in the basal ganglia, and the globus pallidus is a part of the basal ganglia, and the same thing in the basal ganglia, we see this fluctuation that are actually modulated by dopamine. As you remember, the dopamine is the neurotransmitter of the basal ganglia. So there is no doubt that the brain, besides these theta, alpha, delta bands, the brain generate infra-low frequencies. There is no doubt in that. Again, this is the power law function of EEG, full band EEG. As you can see, there are some peaks, and in conventional EEG in alpha frequency band, we have this alpha rhythm, alpha peaks. And when you record this EEG, infra-low frequency EEG, and the other parameter like near-infrared spectroscopy, arterial blood pressure, respiration, you can see that there are different types of low frequencies in these parameters, and the EEG frequency band is actually quite different from the low frequency band in other parameters. For example, arterial blood pressure. We know that the arterial blood pressure oscillates, and the oscillation is about 0.1 hertz. These are the so-called Mayer oscillations. But this Mayer oscillation, they are quite different from the oscillation that are recorded from the surface of the head. And this is a study that was made by Nikulin and co-authors. And they use different parameters, physiological parameters. This is the study, my study from my latest book, the functional neuromarkers of psychiatry. And here is the EEG, infra-low frequency EEG recorded in a subject, in a healthy subject. And I'm not going into detail, but what we see here is that this oscillation, it's actually a traveling wave. You see it travel from CZ electrode position to PZ back and forth. So this is a kind of traveling wave in the brain. And if you compute the CNV wave, so simply you average, you ask the subject to perform a task, at Q, at go, no go a task. And in this task, subject has to prepare after the presentation of the first stimulus of the cue, he has to prepare to the target. And during this preparation, the brain generates negativity, which is called contingent negative variation. And you can see an amplitude. And the topography of this wave is quite different from this spontaneous oscillation. So if you ask me a question, the CNV and what is the relationship between CNV and these spontaneous infra-low frequencies, I would say there is no correlation. So when people work with the method developed by Niels Birnbaumer, this is a kind of discrete slow cortical potentials neurofeedback. This we are doing with completely different measures. They're completely different methodologies. Okay, so first of all, we have when you look at this EEG in the low frequency band, you can see that there are two types of oscillation. First of all, there are rhythmic oscillations, and rhythmicity basically is around 0.1 hertz. It could be a little bit higher, it could be a little bit lower, but the frequency is about 0.1 hertz. And there is also the rhythmic and rhythmic EEG, and here C3, the EEG recording from C3 look like very rhythmic, and EEG recording from the electrodes at P3 looks quite rhythmic. Okay, and here is another example, and this example again shows that the CNV, contingent negative variation, in this particular subject is quite different in amplitude and in topography from that of spontaneous oscillations. And in this case, spontaneous oscillation is about 0.1 hertz. And the CNV has a completely different topography. Okay, so when we worked with these infra-low frequencies of oxygen, we found that the phases are very important. And basically, there are two different phases. One phase is increasing when, for example, the level of oxygen is increasing. we call it increasing phase. And the second phase is decreasing when, for example, the level of oxygen is decreasing. Okay, so and when people they worked with the infra-low EEG, they found this infra-low EEG, the phases of infra-low EEG, they actually correlated with the task performance. For example, if you present a kind of acoustic or somatosensory stimuli on the threshold, so in 50% of cases, the subject cannot detect the signal, and 50% of cases, the subject can detect the signal, and you measure the performance. So there are hits, there are misses. And here is a study that was taken from a quite old study by Monta, 2008. And in which here the blue are hits, the red are misses, omission of the stimuli, and you basically can see that the hits are associated with increasing phases in EEG. And also, this is a study in which these researchers, they recorded not only infra-low frequency, but also the conventional frequency band, like 40 hertz, like 20 hertz, 10 hertz frequency band. And you can see that the power in these different frequency band actually correlated with the increasing and decreasing phases of these infra-slow frequencies. Okay, so this is a kind of a summary of the studies. So we have a rising phase and falling phase of infra-low frequency. And if you measure the behavioral, the performance, that you can see that the hit rate is high on the rising phase of these infra-low frequencies, and is low, the performance is low on the falling phase. The blue line is present a kind of EEG power in different frequency bands. And again, in this different frequency band, the power is high on the rising phase of ESF and low at the falling phase. Also, if you record the so-called galvanic skin response, GSR, the galvanic skin response also usually happens on the rising phase of spontaneous EEG. Okay, I think that here I have to mention that the other method of recording this infra-low frequencies is galvanic skin response, GSR. So you measure the skin resistance between two electrodes placed in different parts of the head. You apply voltage, and the current is proportional to the skin resistance, and the skin resistance actually depends, is very sensitive to changes in arousal, so especially emotional arousal. Remember, I was talking about the modulation inputs from the brainstem, and here I have to mention that the changes in the arousal are associated with changes in the input to the cortex from the brainstem. So these changes modulate the level of cortical activation, and basically they modulate the arousal level of the subject. So the galvanic skin response, they're associated with the arousal changes. This is our studies that was done in 70s, and at the left on the top, you can see this EEG. I mean that the infra-slow oscillation of PO2. PO2 is the concentration of the oxygen by measuring this parameter by polarizographic method. And you can see that the time scale is about, the oscillation is about, the period of oscillation is about 10 hertz. So this is a 0.1 hertz frequency. If you measure the impulse activity of neurons, and this is what shown on the right, then you can see that the dynamic of the neuronal activity is completely different from these oscillations in the oxygen, and they are quite sporadic. But what we found that if you ask the subject to do a task, and the task can change the impulse activity of neurons. So at the middle, you can see that this curve shows the PO2 measured in some part of the cortex. So the changes in impulse activity of neurons, they evoke the oscillation in PO2. And you can see an increase of impulse activity of neurons is associated with the increasing phase of PO2. And we also found that if you measure the phases, the decreasing phase and the decreasing phase of this oscillation, they give you a new window to the brain, to the functioning of this oscillation. So here I present the kind of durations of the decreasing phases of PO2. And this is attention task at the bottom. You see that there's attention task and motor task. As you can see that attention task and motor task, they're quite different tasks, and they produce changes, different dynamic in decreasing phase of PO2. So this actually demonstrates you that the phase is meaningful. The phase of infra-slow oscillation has a quite clear meanings, and the duration of the phase is the very important parameter. Okay. Now let us talk about potential brain sources of EEG ESF. So here I presented a potential sources of EEG ESF. Basically, if you type astrocyte and infra-slow ASF, you will find papers that actually demonstrate that astrocytes, for example, these glial cells, can easily be associated with this oscillation. If you type neuron, impulse activity of neurons, and you will find papers that again demonstrate that ESF, infra-slow frequency oscillation, can be associated with fluctuation in this parameter. Okay, but if you type the autonomic nervous system and say local blood flow, then you also can find a lot of papers that demonstrate that these structures, they can easily generate infra-low frequencies. Okay, so basically we cannot exclude in generation of the ESF, we cannot exclude neither astrocytes, neurons, or arteries. In this case, it's a pile artery in the cortex, which is driving by autonomic nervous system. Remember that we have a kind of autonomic nervous system, and there are two different parts of the autonomic nervous system. One part is the parasympathetic nerves, and they drive, they increase the functioning of the different organs of the body, like heart rate. They increase the heart rate, the heartbeat, and the other automatic nervous system is called sympathetic nervous system, that have a just opposite effect. This is increasing the heartbeat. So one sympathetic is activated, parasympathetic is deactivated, is inhibited. So quite different thing. And if you measure the heart rate variability, so the interplay between sympathetic and parasympathetic nervous system results in infra-low oscillations in the heart rate variability. And you can measure the heart rate variability, you can train the heart rate variability, and basically when you train the heart rate variability, you simply training the interplay between sympathetic and parasympathetic nervous system. But if we talk about the EEG, full beta EEG recorded from the brain, then we're not talking about this sympathetic and parasympathetic system only, but in addition, we're talking about the input of neurons, of glial cells, and of course the local blood flow. And here on the right, I present what happened if you measure the diameter of the pile artery. Okay, so this study was done in, I think, in red. And what they did, they measured the diameter of the pile artery, so it's a blood supply, and they change the neuronal activity in this cortical area. Okay, and a change in neuronal activity causes this kind of response. So first you see dilatation of the pile artery, and then there is a construction of the pile artery. So you see that this simple experiment, they showed you that the blood supply, the changes in the diameter of the pile artery oscillates with the 10 hertz period, so it's 0.1 hertz frequency band. Okay. And in this picture, I didn't show the phenomena associated with galvanic skin response. Okay, so now MRI, bolt signal. So if you look at MRI scan, and that measure basically the local blood flow in the brain and in the resting state. So the subject is simply lying in the scan and with eyes closed condition. In this condition, we have several hot spots in the brain. So one is the medial prefrontal cortex. This is medial prefrontal cortex. The other is posterior cingulate cortex, precuneus. And the third one is angular gyrus. So this is in eyes closed condition. This is hot spots, and these hot spots are heavily correlated. So they form a kind of system, which is called default system, default system of the brain, default mode network. Okay, and here is I schematically present a default mode network in comparison to other network that can be associated with different function, like visual function, like motor function, etc., etc. And if we talk about the ESF fluctuations in EEG, then we are talking about positions of electrodes which are somehow associated with this default mode. And remember, the default mode is associated with the activation in the angular gyrus. So P4 electrode position is basically the position that is associated with angular gyrus. These are the brain region from which you can access to the default mode. So if we talk about the Othmer method, this method targets two primary issues of the regulatory hierarchy. One is brain stability, and the other is arousal regulation. And this interhemispheric placement targets the brain stability, and the parietal placement targets arousal regulation. Okay, and the other statement is that both placement target regions where the default mode network is accessible at the cortical surface. Okay, so that is a kind of a theoretical explanation of why these placements of electrode, why these protocols are used in ESF neurofeedback training in Othmer method. Okay, so here I show you the other study which was made by these Chinese people, and which is called the brain infra-slow activity correlates with arousal levels. Okay, so they use this galvanic skin response, and galvanic skin response is here is presenting in a kind of warm color. I would say it's something is like yellow brownish, whatever, and the cold color is the EEG, infra-slow frequencies in EEG. As you can see, even by looking by naked eye, you can see that there is indeed quite clear relationship between this galvanic skin response and galvanic spin response. I have to tell you, it's a kind of a measure index of arousal level, or cortical arousal level, and ESF. So basically, this study it's indeed shows that the ESF are associated with the galvanic skin response. Okay, then the other study that demonstrate that I think that these oscillations, you can see, they are sporadic oscillations. They are not regular. So, and remember that we were talking about regular and irregular fluctuations in this ESF. So here we are talking about irregular. When we talk about galvanic skin response, we are talking about irregular oscillation. Now let's talk about regular oscillation. Oscillation in 0.1 hertz. Okay, so and this is the study by Rybushinsky. 214, and in this study they were able to measure the directly measuring hemodynamic oscillations, the changes in this in the diameter of this pile vessels, arteries, and they were able to demonstrate that the blood supply, local blood supply, actually oscillate with the 0.1 hertz frequency. Okay, so you can see that if we talk about this ESF, we talk about different sources. So one source could be changes in the arousal level, can be measured by galvanic skin response. The other changes can be associated with blood supply and with local, not systemic. Systemic blood supply is Mayo waves that can be measured by this the blood pressure, changes in the blood pressure, but we are talking about local blood supply, and we are talking about the irregular and irregular oscillations. Okay, so this is, I think that I need to tell you about our studies. I think that our studies was, I think it was a good study. And we have two groups. One group was patients who performs the ELF neurofeedback using the SIGMAT devices, SIGMAT equipment, And the other group was performed high rate, high heart rate variability biofeedback. So it's a different neurofeedback method, and we compared these two groups. So we recorded EEG, the full bed EEG in these two groups. One group performs the real ELF neurofeedback, and the other group performed the HIV biofeedback. And this what remember I told you that if you look at spontaneous EEG oscillation in subject with the brain dysfunction, with cognitive dysfunction, emotional dysfunction, whatever, then you can see the amplitude of this oscillation are quite small. Okay, so here on the right, on the top, you see the average EEG spectra in a group of subject who performs the ELF neurofeedback. So this is the first line, the line on the below is pre-treatment EEG spectra. Okay, and the dotted line is what happened after sessions of neurofeedback, of real neurofeedback. You can see there is decrease of EEG power in and this is grand average, so you don't see these local things because this is a grand average of many subjects. So, and this is a real ESF neurofeedback, changes, increase of amplitude. So basically the ESF normalized in at the baseline, the EEG, the amplitude of EEG is of EEG oscillation fluctuation is quite low, and the ESF neurofeedback increases the EEG power. This is what happened in the control group. You see that this is a group that actually do the similar thing. It does neurobiofeedback. So they're using heart rate variability as biofeedback. And in this case, you see that the EEG power is not increasing. It's just the opposite. It's decreasing, not statistically significant, but there is a slight decrease of EEG power in this. So indeed, the ESF neurofeedback changes in positive way. It's actually kind of normalizes the EEG-SF. Okay. So this is a kind of a summary of what I already told you. I don't think that I need to read this. Basically, that so maybe it's better to repeat. I think, as Jay said, the redundancy is the mother of learning. So the ESF were discovered 60 years ago when the appropriate amplifiers were designed. And to avoid skin-related artifact, the recording required puncturing the skin under the electrodes. Nobody does it anymore. At the beginning of the 21st century, the interest to EEG-SF was renewed by the development of commercially available DC-coupled amplifiers, and by observation of ASF in Bolt signals. We are talking about regular and irregular ASF. So irregular, the independent components of irregular ASF was shown to correlate with the Bolt signal. And these changes in tone are driven by changes in arousal level, which are measured by GSR, galvanic skin response, pupil size, and heart rate variability. There is no consensus regarding the temporal organization of EEG ASF. Indeed, some studies emphasize the absence of peak on the EEG ASF spectra. Some studies report prominent oscillation, as I showed to you. There are quite prominent oscillation in the frequency. In our studies, we found frequency about 0.1 hertz, but there are also studies that they demonstrate the frequency of 0.02 hertz. And some studies emphasize multiple discrete infra-slow oscillation. So I think that this is probably the best way how to describe ESF fluctuations. So they are both there are both oscillations, regular and irregular oscillations. And I actually didn't find any study that the use parameter for PEG-ASF as a neuromarkers to discriminate psychiatric patients from healthy controls. Okay, so I think that Rustam sent me these questions that people ask me to answer, and this is probably a time for answering these questions. But before I will do this, I'm going to hear your questions from the spot. Okay, so please, if you have any questions, go ahead and ask me the question. I'm all ears.
Thank you, Yury. It was amazing excursion into the all these areas that's intriguing us. As you know, we discussed a few times, the Orthmas actually train in the seems like an ultradian rhythm that is now going lower. Do you mind to very briefly say what your opinion about that?
You mean?
They go into ultradian range, even lower than infra-slow activity.
Oh, you know what? I'm a kind of a person who actually try to rely on the facts. Okay, if I look at the spontaneous EEG that is given us in a study, in an experiment, I don't see I don't see any very low frequencies which, to my mind, make any sense. But I cannot exclude that going even down 0.1 hertz frequency give you some advantage. I don't know. I basically don't know because I have no experimental evidence for that. I just can speculate, but I prefer not to speculate. I prefer to rely on the facts. But you know that going even below this frequency point 1 hertz or 0.2 hertz, it's
okay. So you basically already answered the question number 3 from the list. So that's we don't need to cover that.
well, could you make could you make a comparison between marked miss and Moson method?
Yeah.
Well, again, I don't have direct access to these methods, and I can rely only on what is written or was written in the papers. You know, these two groups, they publish quite a lot of papers, and if we talk about Mark Smith method, so basically it looks like they are using amplitude, simply amplitude of ESF oscillations. And they are also using Loretta imaging to localize the sources. So they actually deal with the virtual sources. For example, they can localize the source in the parietal cortex, in the anterior, in the posterior cingulate cortex, and they can actually train these virtual sources using as Loretta imaging. And as far as Othmar method concerns, that I can only tell you that the method based on a kind of clinical facts, thousands, 100,000 of clinical cases, and they're using these clinical cases to adjust the parameters of biofeedback so that the effect would be beneficial for the clients. So it looked like that these two different methods are quite different, and they're quite hard to compare.
Okay.
And I think that the published studies of both methods shows that they indeed work. I demonstrated in this presentation our studies using the Othmar method, and they demonstrated that the method actually quite specific. It indeed changes EEG specifically, and it's quite different from the heart rate variability biofeedback. And a similar thing was shown by Mark Smith group. Remember, I showed you 2018 study in which they compared the training of ESF in the posterior parietal cortex, posterior cingulate cortex in food addicts. So it looked like the two methods, first of all, they're quite different, but they're using different strategies and different sources. But they demonstrate that, indeed, they're beneficial for treatment of patients.
Okay. Great.
That all I can tell, and but quite clear, we don't have a theory behind this. So all this method, they're kind of clinically oriented. this is a quite different approach from that we use in conventional EEG. So in conventional EEG, we more or less know about the mechanism of cortical of this rhythmic oscillation, like we know mechanism of alpha rhythm oscillation. We know that the idling rhythm of the cortex. We know the mechanism of beta rhythm oscillations. So so at least there is some theoretical background that is the basis of conventional biofeedback protocols. But in this case, we don't have any background, any theoretical background. So everything is based on clinical evidence.
Empirical, yeah.
Yeah, and I
Okay.
think that definitely this area, these fields needs a kind of theoretical support. And I was not able to find any studies in literature that tried to compare EEG spectra in the low frequency band of healthy controls in comparison with the different types of categories of patients, psychiatric patients. I wasn't able to find anything like this. So, and this definitely needs to be done.
Yeah.
Okay.
So you already started doing that research in your lab, yeah?
We try, but you know that we quite limited. I mean that we're very thankful to BMedic group that gave us the equipment for free, and we're using this. But you know, still the sources, our main sources, are quite limited because our lab has other topic to study, so that we are getting money from the academy for doing some research, and this research has nothing to do with ESF training. Okay, so the first question: will we ever have a QEEG for ESF? Standardization of ESF assessment? And the answer is probably yes. So remember I showed to you how to do it. So basically you simply, what you need to do, first of all, you need a long recording. So not 2-3 minutes, but maybe 20-30 minutes, maybe more, of EEG, of a full band EEG recording. So the first thing, and you can easily compute the EEG spectra. You can compute the grand averages of the EEG spectra, and you can compare individual EEG spectra with this grand average. So there is no problem of doing this. Okay, so the answer to the first question, yes, probably at some point we will have this standardization of ESF assessment. Will more sensors for source localization or better amps give us better SF signs and better client results? Well, basically, the more electrodes, the better, of course. But for clinical purposes, I still think that 19 electrodes would be sufficient. And I don't think that increasing number will give us something new. I don't think that it's, of course, it will improve localization. The localization ability, but doesn't give a new view to the EEG. Okay, so I answered the third question. Could we ever expect to see the impact of inhibitory neurotransmitters versus vascular versus arousal on baseline ESF? this could be a good study. But if you look at the literature, you basically don't see any studies. So the impact of inhibitory neurotransmitters on EEG, I couldn't find any studies. The impact of vascular, like stroke, traumatic brain injury, migraines, on the baseline ESF, I also couldn't find any studies. There are not, basically, there are not many studies on ESF. There are not many, it's not the mainstream, okay? The mainstream in neuroscience is completely different. So there are a few people who are trying, and I don't think that this study are supported. This study are supported by grants, and this is one of the problems. The question number 5, the connections between CNV and ESF. I think that I already showed to you that they are completely different things. CNV is a kind of electrical response to the associated with expectation, and ESF are spontaneous oscillations. If we are talking about oscillations, spontaneous oscillation, they have quite different amplitude and quite different topography. So there is no connections between these two. At least how in the way how we do it. Okay, given a prediction how to choose to train someone in ASF. That I told you that there is only clinical basis. There is only clinical implications for choosing the ASF. So the selection of ASF protocol is based on simply clinical observation, not on the recording ESF and comparison the EEG spectrum in full band with the database. Nothing like this. Rustam question, you know, Rustam, you're asking me questions that I had difficulty to answer.
Yeah, sorry. we had a discussion about the not really directly related to your presentation. You can ignore that one.
Yeah. Okay. What kind of gel are the best? Well, any gel that have chloride ions are good. So then you have to look at the chloride ions. If the gel has the chloride ion. If not, then you simply can pour some salted water. So pour some salted water in the gel, and then you can be sure that there is chloride ion in gel, and that would be more than enough. Okay. What biofeedback, in addition to awareness, is best for a signal with a time scale as long as 10 seconds?
Classical kind of conditioning theory.
Yeah. Well, that as I mentioned, each company, each group is using its own way of presenting the biofeedback signal. Okay, this biofeedback channel circuit is actually quite differently they use quite different circuits, and they use quite different algorithm for extracting neurofeedback parameters. So then we need to go closer into the way how they doing it. And again, there is no theory, there is no reason how we can judge, oh, this is the best way and this is not the best way. We cannot say this. No theory, and everything is relied on clinical practice, on clinical evidence. And people, first of all, maybe people, they're using different kinds of clients, different kind of patient, and that is why they are driving by different ideas. I don't know. I basically cannot have difficulty answering this question.
Yeah.
Okay, maybe more questions?
There was something not related directly to infra-slow from Miriam was asking about magnetic EEG, ECG resonance therapy. Do you know anything about that?
We're not using it. I mean that when I worked with Ristanatanen, we actually recorded this MEG, magnetic electroencephalogram, and it's very demanding method. It's very demanding method. The equipment is very expensive. And I don't think that this is going to be the case for clinical application. Okay. I see that we're going to stick to stay with these EEG amplifiers and EEG methods, not the.
May I ask something, Yury? Just briefly, just to let you know about new information on this. That's why I asked the question. Are you okay for me? Over the last 4 years, there is very little clinical group who fought very heavily to bring the equipment to Australia of MERT, magnetic EEG, ECG resonance therapy. They managed to do it. It's established north of Brisbane. And over 4 years, they fought and won. So with NDIS and Medicare is funded now. This is extremely expensive, you're right. And it was completely therefore inaccessible for people. But now it's funded by NDIS, and more and more people are referred to this treatment. Apparently very successful in some area of PTSD and ASD, which also depression, addiction, and now they are going expanding it to different clinical presentations. I would love to know and just think maybe put heads together in the future. I don't expect us to have question either of us today, but just to see, because it is funded, we will have a lot of participants actually using this methodology here in Queensland, literally Brisbane area.
Wow, this is a good news. This is a good news. Yeah, if it's funded.
It is funded now by government. Yeah, I have few patients that actually are on that treatment, and I'm really curious to go through that observation and monitoring. You know, of difference.
The only different when you use MEG, you are not dealing with this kind of volume conductance. Okay. In EEG, the volume conductance is a big problem, because when you record EEG in one place, from one electrode position, the same signal can be measured from different electrode position. But if you are using MEG, you are locally you are localizing the source of the signal. So localizing the source is very important issue. So maybe the image is has a kind of advantage.
Yeah, Yury, I went to their presentation, and I think I think they just it's a fancy name for EEG based magnetic resonance treatment that many centers now do.
Oh, you mean and this is a difference? Okay.
Yeah, it's just not TMS. It's just TMS method. It's just a fancy name that's confusing, but they're using some kind of decoder, some resonance, EEG resonance. They're basically guided by EEG. One of our clients went to America and had the treatment there. They identified that the autistic kid hasn't got alpha, so they were pushing alpha up. That's what they were doing.
Well, that kid tremendously improved, because I know the patient, and he's now going to Rudolf Steiner School, and he was completely non-reciprocal 7 years ago. That's the mother who actually pushed all of this to come, and you know, she was bringing it to you, and you were very, very helpful. But at some point, his progress plateaued. When he was 4, he's now 10. And he's really functioning well. So what I haven't yet gone on to observing exact what is underneath the protocol, but curious to see what's happening, because some of the clients have amazing improvements, some of them have none. And, you know, of course, like in every new protocol, this is totally new protocol, I understand. I wouldn't sort of doubt much, because everything is doubtable. Everything new is always having a lot of nappies in the development stages of the protocol.
But if you look at the physics of it, the magnetic stimulation propagates a signal, so it can inhibit or stimulate the signal. Actually, I've got Tim Rolf here. He probably can comment on it. He uses TMS, and so there was a lot of researchers doing this, and with amazing, like a lot of success with depression. You know.
That's why PTSD as well, you know.
And bipolar as well.
Bipolar as well. A lot of other things now, they are going wider and wider in clinical presentation. I'm really grateful for Yury, for your explanations, that one aspect of sort of advantage we can say is that no issues with volume conductions. So signal source is localized, and it's actually very logical. Thank you for that. So it's one of how, you know, sort of what to look for. Tim, you I know that you unmuted yourself.
No, thanks, Rustam. I just looked at their website. I think there's a bit of confusion. I don't think it's MEG. It's not magnetic EEG, like the magnetic EEG that Yury is talking about. Basically, what they're doing is using the EEG and the QEEG specifically to guide a TMS prescription. Right. So the actual intervention is transcranial magnetic stimulation, which is very widespread, and then they're just combining that with an EEG for assessment. And so it's not really revolutionary at all in many ways. I mean, I do this kind of thing. I think a lot of people do it, and there's a lot of literature actually that's looked at are there predictors in the EEG that can help you to personalize TMS interventions. You know, we won't go down that pathway, but there is quite a lot. So I suspect that it's just a good marketing exercise. I'm not saying TMS isn't a good intervention. It is potentially a very good intervention in certain circumstances. But yeah, so
Yeah,
sorry to
Okay, so
interrupt.
is that what you found out? Okay.
Yuri, we can see your mail.
You mean you don't see my face or what?
Oh, yeah,
Yeah.
that's right.
We lost you.
Yeah, I can see it. And
Okay.
something happened, you know, with this with the, well, but I believe me, I am with you.
Okay, great. Well, thank you so much. It's really amazing how you pulled so much data together, because a lot of controversy and people seems
Yeah.
don't trust the new methodology, and this will be really help to educate and our field to embrace the method of infra-low frequency training, infra-slow fluctuations training. So as you know, we've been using that for the last 11 years, and you've seen pre and post changes sometimes. Yeah. All right. Well, probably we should finalize now. Yeah, if no other questions.
Just to
I
say
asked,
thank you very much.
sorry,
Yeah.
I just want to say thank you as well, and want to ask to really think about some long, good, thorough training, 1 or 2 week training next year or soon. You know, would be
Yeah.
really lovely, because we really feel always when I listen to any of our presentations, especially by Yury, I feel how little we know from Yury. Yeah. So
Yeah. Well, we'll
would be
probably
really
That is-
great.
That is easy. That it can be arranged. Rustam?
Yes, we'll definitely will organize something. We just have to wait for the
Good.
situation to settle with visas. Yeah. At the moment, we have to check all the situation with visa, and we'll start planning the workshop.
Yeah.
Yeah. Thank you so
Okay.
much, Yuri.
Thank you. Thank
Thanks
you very
everybody
much.
for attending.
Your master,
Okay.
and good luck. Good luck, and remember that we don't know many things in this field, but we are trying to find out what is the best way. And we are actually quite limited. Remember that it's not a hot topic in neuroscience. Okay, so good luck to all of you.
Thank you.
Thank
Yuri.
you.
Thank you
you.
too. All the best.
Thank you very much.
See you
Bye.
soon.
Bye.
Bye-bye.
See you.
Bye, everybody.
Yeah.